Hello viewers, welcome to this particular lecture on microbiology. In this particular lecture, I shall be covering some aspects of genetics with particular reference to Mendelian genetics and of course post-Mendelian development in genetics. To start with, I shall be giving some historical background. Actually, Gregor Johann Mendel was a clergyman from present-day Czechoslovakia, at that time it belonged to Austria, who had his basic degree in statistics and mathematics. But he was a priest by profession, and in a monastery in Brunn, he actually did some unique experiments in the garden of the church, where he utilized some pea plants, to be specific garden pea or Pisum sativum, for his experiment. And these experimental results were printed in a particular paper published by the Brunn Natural History Society Proceedings, and the title of the paper was Versuche über Pflanzenhybriden, or Experiments on Plant Hybridization. Now, it must be very clear that these particular results on pea plant were on the genetic principles at a time when chromosome, gene, these were all unknown. So definitely this discovery was ahead of time. But the question is, why did he select pea plant as his experimental tool? This is because pea plant is an annual plant where there are 7 pairs of contrasting characters with clear expression of each of these characters. That included the seed shape, where spherical dominated over wrinkled seed color which is yellow and green, flower color which is purple or pink dominating over white, and pod shape which is inflated and constricted as the dominant and recessive character. Pod color green is dominating over yellow. The flower position, that is the apical flower, is recessive to axial flower which is dominant, and at the same time the stem height, where tall pea plant is dominating over dwarf. So these particular characters were qualitative characters which actually Mendel selected because these characters were clearly expressed on one side, and on the other side the pea plant can easily be hybridized because their pollen grains were bigger in size, and you can easily produce a progeny with all these characters. And at the same time, the first law which he stated was the law of dominance, meaning if there are a pair of contrasting character, then the dominant character will be dominant over the recessive character, and in a cross the dominant character will be expressed but not the recessive character. The second law basically is actually called the law of segregation. This particular law states that when a particular tall and a dwarf pea plant are crossed, he called it factor because gene was not known at that time. In the F1 or first filial generation, all the plants were tall, and in this case the tall character was dominating over dwarf, and when two such hybrid tall, as he called it, were crossed, in the second filial generation 3 were tall, 1 was dwarf. So this ratio where 3 are dominant tall, they were called the phenotypic ratio, meaning 3:1, and the genotypic ratio is 1:2:1, where 1 is the pure tall, 2 are hybrid tall, 1 is pure dwarf. So on the basis of this single pair of contrasting character, which is also called monohybrid cross, Mendel stated that when two different factors are crossed to each other, initially they come close to each other, but at the time of gamete formation they segregate from each other, expressing in the ratio of 3:1. Coming to the second law, which is based on dihybrid cross, where he clearly showed that when there are two pairs of contrasting character, meaning the yellow cotyledon dominating over green and smooth cotyledon dominating over wrinkled, it was shown that when yellow round is crossed with smooth or yellow smooth is crossed with green wrinkled, in the F1 generation all were hybrid round and yellow. But when two such hybrid were crossed, it clearly produced 16 different individual, of which 9 were yellow round, 3 were green round, 3 were yellow wrinkled, and 1 was only green wrinkled as the pure recessive. So on the basis of this experiment, he said that the characters not only segregate, that is the yellow round is segregating from the green wrinkled from each other, but they also produced intermediate forms. What are they? The intermediate forms were the green round and the yellow wrinkled, which were not there in the parent. And this particular law was known as the law of independent assortment, and it is a typical characteristic feature of dihybrid cross. So this particular experiment actually is a very important step to understand variation in a given organism. And actually this law of dominance and segregation is considered as the first law, and the law of independent assortment is considered as the second law, where this particular law had a ratio of 9:3:3:1 as the phenotypic ratio. And when we consider the present day cell division, actually the first law is clearly indicated by the anaphasic separation of chromosome during gamete formation in meiosis, and the second law is explained by the exchange of chromatid segments by crossing over during the Pachytene substage of meiotic prophase 1. So you can understand that this particular discovery was done at a time when cell division, chromosome, the ideas were not clear. And this particular independent assortment has got a tremendous impact in the variation of different organisms. Why? Because say for example human gametes with 23 pairs of chromosome, the number of possibilities will be 2 to the power 23, huge number of possible combinations, which indicated that one gamete will be different from the other, and thereby it accounts for the variation within human population. That is, not even two brothers, they are not the same, they don't look the same because of this crossing over phenomenon causing independent assortment. So there lies the success of Mendel. But as you see, after the discovery, people were all criticizing Mendel. Why? Now he clearly stated that this particular feature is exhibited by pea plant. But it was Carl von Nageli who showed that Hieracium is a plant which is belonging to family Asteraceae, is not exhibiting this particular trait, which is 1:2:1 or 9:3:3:1 that was described by Mendel. And Mendel actually again did the same experiment in Hieracium, but he was genuine enough to show, well, it is not showing the same trait. And there lies the problem. When Mendel stated this particular result in the same journal, people started disregarding Mendel. He was actually he became a forgotten hero, and he died of total ignorance, ill health in the year 1884. But it was after 16 years of Mendel's death, 3 different scientists, namely Hugo de Vries, Karl Korrens and Erich von Schemack, from 3 different parts of the world independently proved that Mendelian ratio holds good in 3 different plants. And they didn't stop here. They made the republication of Mendel's discovery in a journal called Flora. So this particular event is termed as rediscovery of Mendelism. And Mendel was considered to be the father of genetics. At this particular point, when Mendel got his credit, he was not there in this world. So he still remains an unsung hero, though his discovery was ahead of time. And this particular discovery of Mendel, when people understood the importance of his discovery, several other points came into existence. It was given by Bateson. Bateson, actually in the year 1903, he coined the word genetics, he coined the word allele, meaning that the factors which are showing the two contrasting character. Subsequently, there were other important discoveries by Thomas Hunt Morgan, who worked in Drosophila, and he showed that this particular gene expression actually occurs through chromosome. So chromosomal pattern of inheritance was discovered based on Mendelism. And another unique success of this particular Mendelian work is because of the fact that Mendel did all these experiments on the basis of mathematical data, and this data, this is actually his manuscript. You can see that he worked on mathematical data and he calculated the results on the basis of statistical analysis. So when a particular breeding result is done on the basis of huge number of data properly statistically analyzed, it cannot be wrong. And at the same time, he considered characters which are qualitative character, which is clearly expressed by the pea plant. So that is another point of success. And if we consider the present day traits in human, certain diseases like the sickle cell anemia, which is because of one particular gene variation where the RBC becomes sickle shaped, or the symptoms of xeroderma pigmentosa with some typical pigmented spot on the skin, it is clearly following Mendelian trait. So there lies the success of Mendel. After so many years, we still follow Mendelism. But that doesn't mean that Mendel actually was stating everything to be followed by each and every individual. There are certain discoveries which were made after Mendelism which showed that these discoveries will clearly deviate from Mendelism, and they are called post-Mendelian inheritance or post-Mendelian genetics. One such important discovery was the discovery of linkage. Linkage was discovered by Bateson and Punnett, after whose name there is the Punnett square or the checkerboard. What did they observe? They observed that the parental characters in, say for example, the pollen grain of pea, which is purple and long, dominating over red and round. Normally, it is expected following the 9:3:3:1 ratio, there will be more parental types and there will be equal number, that is almost 3 parts of individual plants with the mixing of the contrasting character. Meaning, if purple long and red round are the two parents, then there will be purple round and red long. It was observed that the parental forms were more, and to be specific, when the test cross was done, it was observed that test cross, meaning the crossing with the homozygous recessive parent, it should have produced 1:1:1:1. Instead of that, they produced 7:1:1:7, meaning that the parental characteristics have not separated as because they were linked. Another important discovery was the incomplete dominance. As per complete dominance, when a red colored flower is crossed with white, in the F2 generation, it should have produced 3 red and 1 white. But in incomplete dominance, because the red is incompletely dominant over white, in Mirabilis jalapa or 4 o'clock plant, there will be 1 red, 2 pink and 1 white. What does it mean? It means 1:2:1 will be both the phenotypic ratio and also the genotypic ratio. Another important character is codominance found in this typical Rouen variety cattle. What is that? A particular red variety is crossed with white. We should have got 3 red and 1 white, but because it was codominant, some red characters will come in at certain specific location of the cattle. So it will give rise to Rouen variety, and it will be 1 red, 2 Rouen, 1 white. So it was codominance in animals, in cattle. Similarly, there may be important traits like blood group inheritance. What does it mean? Blood group normally will have typical multiple allele, meaning one particular phenotype will be controlled by at least 4 to 5 different allele, and this particular allele will combine with each other, and that will give rise to the resultant blood group. And that is the reason why globally certain blood groups are more popular, more common, like O blood group, or in this part of the world like the B blood group. Similarly, polygenic trait is another important trait found in case of human or in case of Drosophila eye color, where we see clearly that a particular character is controlled by more than one gene or one pair of genes. The example of lethal gene in rat is another unique example where the yellow coat color in pure condition is causing death. So a cross between two hybrid yellow will produce two hybrid yellow and one white, and the dominant yellow will die because this is the dominant lethal gene. So the ratio becomes 2:1 instead of 1:2:1. In case of certain organisms, like say for example some cats or in some other lower mammals or in some plants, there may be the recessive lethal gene where there will be the dominant and the two hybrids surviving, but the recessive lethal individual will die. So the ratio becomes 1:2 for both phenotype as well as genotype. Another very important discovery is the discovery of genetic interaction or epistasis. What was Mendel's view? Mendel's view was one dominant gene, one recessive gene. Always the dominant gene will be dominating over the recessive, and that means the interaction takes place between these two particular genes at the same locus on the homologous chromosome. But actually what happened was, in case of epistasis, that two adjacent genes will interact with each other, and that actually will tell upon the dominant recessive relationship of one pair of genes, as observed in case of the comb pattern in poultry bird, where the cross between rose comb, this is the rose comb, and the pea comb, this is the pea comb, should have produced the rose comb, because if the rose was dominant, but there was some interaction, and instead a third character came up which is all walnut. This is the feature which came up. It will not end here. When two such walnut combs were crossed in F2, there were 9 walnut, 3 rose, 3 pea and 1 single. This is the single variety. So what does it mean? It means that the characters do not have this dominant recessive interaction, but at the same time they are interacting with each other when they are in adjacent position. And this particular feature was taken up, and a series of such epistasis was observed. One such epistasis was the dominant epistasis, meaning the epistatic gene is dominant gene, and that modified the ratio 12:3:1 found in case of dog, meaning the dominant character will be the white here 12, there will be the 3 yellow color where there will be one only single dominant gene, and the only one recessive gene will have the single individual. Similarly, it can be the recessive epistasis found in case of rat. What was there? It was found out that a particular quality of rat is agouti, where there are both A and B. But when this A and B were not there, that means only either capital A is there either in pure form or in hybrid form, it will produce the black color. But if this capital A is not there, it will be the recessive epistatic gene of small a which will make the rat albino. So the ratio of 9:3:3:1 is modified as 9:3:4. Another case was the dominant recessive epistasis, meaning a pair of dominant recessive character and also a recessive character will express white color in case of flower of dicot, or in case of leghorn, meaning the poultry bird. What happened was there were 13 white and 3 colored. Why colored? Because this particular colored pattern only had this one single dominant gene, and this particular dominant gene had made it colored. All the others became white. This is another important case which is actually called the duplicate gene. Duplicate genes may have different pattern, they may have some cumulative effect, as seen in case of the shape of the chloroplast in case of dicot plant, where the ratio became 9:6:1, where 9 is the dominant disc-shaped chloroplast, 3 and 3 meaning only with one single dominant gene will have both spherical shape, so it became 6, and only one long which is without any dominant gene. So the ratio is showing a typical cumulative action and the duplicate gene effect of either capital A or capital B. This is another important case where we see that it is duplicate dominant epistasis, meaning whenever there is some dominant gene present, say for example either capital A or capital B, if they are present, they will all bring about the typical triangular character of the ovary, but if it is absent it become round. So it is quite evident that any one of the dominant gene will be present in any one of the particular individual, so the ratio becomes 15:1. And this type of character is normally found in case of wheat grain, where the typical kernel color, that is the golden brown color, is dominant. Hardly we find any white colored grain. Another important discovery is the duplicate recessive epistasis, where the recessive epistatic gene is present and it will have a duplication effect, meaning the gene will complement each other. How? Say for example in lathyrus, which is sweet pea, the petal color is controlled by a pigment called chromogen, and there is a particular enzyme that produces the chromogen. So if both the enzyme producing gene and the petal color are present together, then the colored petal or the purple color will come. And they are present in 9 different individuals and absent in the other 7. So the ratio becomes 9:7, or 9 purple is to 7 white. As because these genes are complementing each other, it is called complementary gene. So thereby we can see that these different cases of epistasis or the different cases of the linkage or incomplete dominance or codominance or lethal gene or sublethal gene was not discovered at the time of the discovery of Mendelism, which were discovered at a later time, and they are all correlated under the different development or post-Mendelian genetics. In conclusion, we can say that epistatic can be of different types, which can be dominant epistasis, recessive epistasis, duplicate epistasis, duplicate recessive and dominant recessive epistasis. And we can definitely understand this much, that Mendelian discovery was a discovery ahead of time, because at that time chromosome, genes, nothing was known. Being a mathematician, he actually produced a set of results which are statistically significant and correct, of course. It was not his fault that certain discoveries were made much later. And of course, at the same time he was lucky, because the genes were not linked, they were not showing incomplete dominant, neither they showed any codominant, and at the same time they were actually free from any type of epistatic effect or epistatic genetic effect. So thereby we can understand that Mendelian principle still holds good in different group of organisms, though after Mendelism lot of new things have been discovered which should be considered under the post-Mendelian discoveries of genetics. Thank you.